Hello, everyone. For those of you who don't know me, my name is Briana. I'm a third-year graduate student in Roberta Brambilla's lab. And today I'm going to be talking about my project, where I'm looking at the role of Astroglial TNFR2 signaling in synaptic and cognitive function in multiple sclerosis. So to start out with some background on multiple sclerosis, or MS, MS is a chronic neurodegenerative disorder that affects more than 2.5 million people worldwide. It's a T-cell mediated autoimmune demyelinating disorder, and the main hallmarks include acute inflammation, oligodendrocyte death, demyelination, axonal degeneration, and neuronal loss. So MS manifests with distinct clinical phenotypes. A majority of patients are diagnosed with relapsing remitting MS, and this is characterized by periods of increased symptoms and disability, which is then followed by periods where the patient can either recover completely or plateau before another relapse occurs. And about 50% of these patients then go on to develop secondary progressive MS. So this is characterized by the gradual worsening of disability and symptoms over time. So basically, as the disease progresses, those periods of remission begin to stop and the disability just gets worse and worse. And then the rarest MS phenotype is primary progressive MS. This is about 15% of cases, and this is characterized by the gradual worsening of disability and symptoms from onset of disease. So in this phenotype, there are no periods of remission. So despite these differences in clinical phenotypes, all of them are characterized by a variety of different sensory motor deficits. And this includes anything from walking difficulty to spasticity, numbness, fatigue. There really can be a lot of type of symptoms that these patients can experience. But most patients experience some sort of cognitive dysfunction. So about 60 to 70% of MS patients experience a wide range of cognitive impairments. And so what this chart is showing is that out of the patients that experience cognitive impairments, these are the most common types. So for example, memory loss, problems with information processing speed, verbal fluency, concentration or attention span, executive functions, and visual spatial functions. And cognitive deficits are not necessarily dependent on how severe the disease is. And they're not dependent on axonal degeneration or demyelination and different stuff like that. So for example, a progressive patient who has very advanced neurodegeneration may experience little to no cognitive deficits, while a patient that has very little neurodegeneration may face no cognitive deficits or very pronounced cognitive deficits. And so MS is a disease that manifests pretty early in age, around 20 to 40 years of age. And this is really a time in people's lives when they're entering the workforce and they're developing professional and personal relationships with people. And so having these cognitive deficits really represents a major burden. Mm-hmm. No, I think I misspoke there. So it's really widespread. It can affect a patient that has advanced neurodegeneration, they may present no cognitive deficits, but a patient that has very little neurodegeneration may have pronounced cognitive decline. So it doesn't necessarily depend on how severe the disease is. Oh. No, cognitive deficits, they're not dependent on where the demyelination is occurring. Yeah. Okay, so we know that cognition is dependent on proper synaptic function, and it used to be thought that neurons were the only participants in synaptic function. But we now know that at the synapse, pre- and postsynaptic neurons interact with astrocytes in a bidirectional fashion, making up what's called the tripartite synapse. So at this tripartite synapse, astrocytes play a major role. They can regulate synaptogenesis, synaptic plasticity and stability, and the efficiency of synaptic transmission. And they can do this via glutamate buffering. They can modulate glial transmission, and they can aid in synaptic formation and elimination. So synapses have the ability to strengthen or weaken over time in responses to increases or decreases in their activity. And this is a concept referred to as synaptic plasticity. And one of the main forms of synaptic plasticity is long-term potentiation, or LTP. And so this is the persistent strengthening of synapses over time in responses to increases in activity. And this occurs when brief high-frequency trains of activity from a presynaptic cell triggers a large depolarization of the postsynaptic cell, and this produces an increase in the efficacy of synaptic transmission. And so one of the main places that this occurs is in the hippocampus between the CA3 and the CA1 region, and what's called the Schaefer collateral synapse. Okay. So a key modulator of synaptic plasticity is tumor necrosis factor, or TNF. TNF is an immunomodulatory cytokine that regulates inflammation, cancer, autoimmunity, and infections. And it exists in two forms: a transmembrane form that functions via cell-to-cell contact, and a soluble form that's generated via the cleavage of transmembrane TNF by tumor necrosis factor converting enzyme. And so TNF can bind to two receptors, TNFR1 and TNFR2, and these receptors differ in their ligand affinity, their cellular expression, and their downstream signaling. So in neurological disease conditions, for the most part, soluble TNF signaling occurs via TNFR1 and is detrimental, leading to apoptosis and inflammation, while transmembrane TNF signaling occurs via TNFR2 and is protective, leading to things like cell survival, anti-inflammation, and remyelination. And like I mentioned, TNF plays a role in synaptic function. It can strengthen glutamatergic synaptic activity, it can alter synaptic scaling, and it modulates synaptic plasticity. And it does these things by acting on TNFR1 receptors on astrocytes, which really highlights the importance of astrocytic TNF signaling. And so in neurological disease, the level of TNF increases, and its role turns from a regulatory role to either a neurotoxic or neuroprotective role. And this increase is seen in MS, where in the cerebral spinal fluid and serum, TNF levels are elevated compared to unaffected individuals. And TNF levels are increased also at the site of active lesions. And these levels correlate with the severity of lesions as well as the disability score. So as that increases, you can see that the level of TNF also increases. And it also correlates with the disease progression. So as the disease progresses, the level of TNF also increases. So basically, in neuroinflammatory conditions, the interaction between astrocytes and neurons is disturbed at that tripartite synapse. And so therefore, astrocytic TNF signaling is disturbed, and this can lead to synaptic impairment and therefore cognitive dysfunction. And so this was really shown in a key paper by Habas in 2015, where they found a causal role of astrocyte TNF signaling in the hippocampal synaptic alterations and cognitive impairment induced by an EAE animal model of multiple sclerosis. So basically what they did in this study was that they ablated TNFR1 from all cell types, and then they re-expressed it so it was only expressed in astrocytes. So that group is shown in the red. And when they did this, and then they induced EAE, they saw that there was an increase in excitatory signaling in this TNFR1 re-expression group. And this correlated with cognitive deficits that were observed in a fear conditioning test. So these mice that had TNFR1 re-expressed in astrocytes exhibited less freezing behavior, which indicated that they had a cognitive deficit in associative learning. So this was really important because it showed that while soluble TNF to TNFR1 signaling in physiological conditions is necessary for proper synaptic function, in neuroinflammatory disease conditions such as EAE and MS, it becomes overactivated and it can lead to synaptic impairment and therefore cognitive dysfunction. And so despite the fact that TNFR2 is also expressed in astrocytes, and it's been shown to play many protective and reparative processes in EAE, no one has ever looked at the role of astrocyte TNFR2 signaling in physiological conditions or in EAE and MS. So this led us to first ask the question of, what role does astroglial TNFR2 play in modulating synaptic and cognitive function in physiological conditions? And so to answer this, our lab created a loss of function model where we have the conditional ablation of TNFR2 in astrocytes. So in this model, we have our astrocyte-specific promoter, GFAP, bound to Cre recombinase, and this is crossed with a mouse that has TNFR2 in between two LOX-P sites. So this results in a double transgenic that when we inject with tamoxifen binds to the estrogen receptor on Cre, and Cre will translocate into the nucleus and ablate TNFR2. So to first investigate this, our previous postdoc Placido wanted to perform these key behavioral tests that are hippocampal dependent in the physiological conditions. And so he went ahead and did Morris-Water maze and novel object recognition tests in males. And I've also added on to this data by performing the Morris-Water maze test in females. And so the Morris-Water maze test is a test that is looking at spatial learning and memory. And so basically a mouse is placed in a pool and there's 4 visual cues on each side of the pool and then a hidden platform on one side. And so during the acquisition phase, the amount of time the mouse takes to reach that hidden platform is quantified as a measure of spatial learning. And then during the test day or the probe trial, that hidden platform is removed and the amount of time the mouse takes to reach that hidden platform area is quantified as a measure of spatial memory. And so what we found is that when we ablated Astroglial TNFR2, these mice, both females and males, took a longer amount of time to find that hidden platform area compared to controls. So this indicated to us that ablation of Astroglial TNFR2 results in impaired spatial memory and physiological conditions. And so Placido also went on to perform the novel object recognition test, which is a test looking more at long-term memory. And it's based off the idea that a mouse wants to investigate a novel rather than a familiar object. And so what he found is that in male mice, the knockouts shown here in the white circles they had a harder time discriminating the novel from the familiar object, indicating that they had impaired long-term memory compared to controls. However, he didn't see this same effect in the females. So this indicated to us that maybe there could be a potential sex difference. But as I mentioned, he had first just done these studies in males. So because he only saw this cognitive phenotype in males, he went on to do the rest of the preliminary studies in male mice. And so, yeah.
Males at first assessment, day 1, and the water maze, it looks like there's almost completely different inverse effect.
Yeah, it's kind of hard to gather knowledge from the training phase, just because it's very variable and you don't know if the mouse is actually taking the correct path to the platform. And just based on a lot of literature I've looked at, it's really hard to draw conclusions from the training phase. It's more of the probe trial that we can really draw the conclusions from, because you can see the females, there's no significant difference in the training phase, but there is in the probe trial. Okay. So he then went on to look at LTP, and he found that in the knockouts, there was a suppression of LTP compared to the controls. And this was also accompanied by alterations in synaptic proteins. So for example, in SNAP25 and syntaxin 1B, which are synaptic proteins part of the snare complex, there was an increase in the knockouts compared to the controls. And then he also looked at gliosis. And so if you look in this figure here, you can see in green is GFAP positive astrocytes, and in red is IBA1 positive microglia. And you can see that in the knockouts compared to the controls, there are more astrocytes and microglia. And so he validated this with seriology, where he counted these cells in the CA1 and the CA3 region of the hippocampus. And he found that there were more of these cells in the knockouts compared to the controls. So this also indicated to us that ablation of astroglial TNFR2 leads to hippocampal gliosis. So this data, combined with other data from our lab showing that TNFR2 plays a protective role in EAE, led me to my hypothesis, which is that astroglial TNFR2 plays a protective role in containing or counteracting synaptic and cognitive dysfunctions associated with EAE. So EAE, or experimental autoimmune encephalomyelitis, is the most commonly used animal model of MS. And it basically causes the breakdown of the blood-brain barrier and an immune response to myelin-specific antigens. And this is followed by peripheral immune cells infiltrating into the CNS. And so this results in an actual locomotor phenotype, which we score on a scale from 0 to 6. So 0 being a completely healthy mouse, 6 being death due to the disease, and then different levels in between. So for example, 1 is a loss of tail tone, and then the paralysis kind of starts to move towards the front of the mouse. So for example, a 4 would be complete hind and forelimb paralysis. And so how this model works is we start out on day 0 with a pertussis toxin injection. And what this does is it begins to break down the blood-brain barrier. And then on day 1, we do a myelin oligodendrocyte glycoprotein injection. And this specific peptide sequence from MOG exposes these peripheral immune cells to protein expressed in myelin. So then on day 2, when we do another produces toxin injection, these immune cells that were primed against MOG will infiltrate and they'll attack myelin. And this mimics what happens in MS. So we start giving the animals their daily score around day 8 because we want to catch the onset of disease, which is usually from day 12 to 16 in our model. And so we consider the acute phase to be from onset of disease to about day 25. And in this phase, the scores are mainly driven by the infiltration of peripheral immune cells, which is T cell mediated. And then the chronic phase, which is about from day 25 throughout the rest of the timeline, the scores here are mainly driven by neuronal damage and demyelination. So although these phases aren't black and white on what's happening, this is like a main characterization of what is mostly happening during these phases. So this brings me to my specific aims. And for my aim 1, I want to determine the role of astroglial TNFR2 in cognitive function and hippocampal synaptic plasticity following EAE. And I hypothesize that astroglial TNFR2 activation is needed to counteract synaptic and cognitive dysfunctions following EAE. And so I first started out by doing these behavioral tests that Placido and I did in the naive conditions in EAE. And a lot of my first work was trying to just figure out the best test for these EAE mice. So I first started with the Morris water maze test. And although I only included mice that were able to swim, so they had a clinical score of a 2.5 or less, I noticed that just the fatigue and lack of motivation that these mice had just was making it hard to include them. And so I decided to move on to the novel object recognition test. So like I mentioned before, this is a test that is looking at long-term memory. And it's based off the idea that a mouse wants to investigate a novel rather than a familiar object. So we basically start out day 1, just habituating the mice to the box. And then on day 2, there's 2 objects placed on each side. They're identical objects. And then on day 3, one of those objects is replaced with a new novel object. And so basically the amount of time the mouse spends at the familiar object is subtracted from the amount of time the mouse spends at the novel object. And then we divide that by the amount of time spent at both objects, which gives us the discrimination index, or how well a mouse is able to discriminate the novel from the familiar object. And so I started these studies in female mice. So all this data that I'm showing in EAE is female mice. I do have male studies going on as well, but I wanted to begin with the female mice. And so like I showed before, in the naive conditions, there was no differences between the 2 groups. And then I went on to look at pre-symptomatic EAE. So this is 10 DPI, and this is after the disease has been induced, but before the mouse is showing any locomotor symptoms. And so I also didn't see any differences between the two groups in pre-symptomatic EAE. But then when I did chronic EAE, I saw that the knockouts seemed to have a trend where they had a harder amount of time discriminating the novel from the familiar object. And so this indicated to us that astroglial TNFR2 ablation has this trend where there's worse long-term memory at chronic EAE. So I then went on to do the fear conditioning tests, and I found that this is the best test for these EAE mice because it doesn't require them to move around so much. And this is a test that's much more hippocampal dependent and less variable than the novel object test, which sometimes is hard to pull out differences. And so far I've been able to do naive and pre-symptomatic EAE. And what I found so far is that for both naive and pre-symptomatic, we don't see any differences between the 2 groups. And so I'm actually in the process of doing the chronic time point now, and I'm definitely interested to see if we see that same cognitive deficit or that trend of a cognitive deficit we saw in the novel object in the fear conditioning. So that will definitely be interesting to see. But I think maybe one of the reasons why we're not seeing any differences in pre-symptomatic EAE is the infiltration of those immune cells, which is causing this big neuroinflammatory reaction and maybe overshadowing the effects of astroglial TNFR2. So in addition to the behavior for each day of this timeline, I give the mice a clinical score, which I explained we score on a scale from 0 to 6. And so what this is showing is the clinical score for these mice from day 0 to day 40 of the study. And then on the right here is the same graph, but it's graphed from days from onset. So each of these mice have a different day of onset. And so this is a nice way to visualize all their paths of sickness altogether. And so what I found is that it seems the onset of disease and peak disease, there doesn't seem to be a difference between the two groups. But then chronically, there's this separation where the knockouts have a worse clinical outcome compared to the controls. And this is actually significant in the days from onset using the Mann-Whitney test, which is just comparing the means of the two groups. So this is an interesting result, but the clinical score is mainly like it's mainly due to the infiltration of immune cells into the spinal cord. And since I'm more focused on the cognitive function, this is not necessarily the focus of my project, but it definitely is interesting, and it indicates that maybe astrocytes lacking TNFR2 become more inflammatory, or they're involved in more processes of axonal injury and demyelination. So one of the things I wanted to see is that if the disease score correlates with the cognitive score. And so what I did is I looked at the correlation between the score on the day of the test and the discrimination index for the novel object test. So that's shown here on the left. And then on the right, I wanted to look at the correlation between the cumulative disease index, which is the sum of all the scores, and the discrimination index. And so I found that the disease score does not correlate with the cognitive outcome. So it doesn't mean that the more sick the mouse is, the worse their cognitive outcome is. So this was definitely interesting, and it shows that there's probably two different mechanisms taking place, one that's producing locomotor deficits and one that's producing cognitive deficits. Yeah.
I was looking at it, it doesn't really necessarily evaluate spinal functions. All those scores would be due to something happening at the level of the spinal cord.
Exactly. Yeah. Yeah.
I got to ask you finally. I mean, I think
yeah.
But,
no problem.
have I ever seen about this picture of the campus and these mice? Look at any evidence that you see demyelination in different campuses will come up. Okay.
Yeah, I mean, there.
Show that so that might feel activation. Seeing the evidence of demyelination in campus or cortex, I'll take that as well.
Yeah, so I haven't looked at that, but this is something that we're definitely interested in. And I one of the things I wanted to know is if immune cells can infiltrate into the brain. And yeah. So I haven't seen any evidence of the demyelination, but in the literature, it has been shown that there could be demyelination. It's just not as intense as what happens in the spinal cord. Yeah. Yeah. Okay. So these studies with the loss of function animals basically brought up the question that if the ablation of astroglial TNFR2 leads to a worsening of cognitive function in EAE, does upregulating astroglial TNFR2 improve cognition and basically rescue this phenotype? And so what we did is our lab created a gain of function model. This is an inducible TET-off model where we have our astrocyte specific promoter bound to a tetracycline transactivator. And in the presence of doxycycline, it's inactive. But when we remove doxycycline from the food, it can become active and it can bind to the tetracycline response element, where it will increase the transcription of TNFR2. And this will lead to TNFR2 overexpression. So far with this model, I started out doing the fear conditioning test because I found that this is the best test for these EAE mice. And I really wanted to focus on the chronic time point because I saw that trend of deficits in the novel object test with the knockouts. And so although I don't have as many animals for these groups because it's been hard with the breeding to get these double transgenics, so far for the naive conditions, we see that there doesn't seem to be a difference between the freezing behavior in the naive conditions. But then at chronic EAE, we see this increase in freezing behavior in the overexpressor mice compared to the control mice. So these overexpressor mice freeze more, indicating that they've learned to associate this the apparatus with the shock that they received during the training phase. And so the context, I think I forgot to mention this, but the contextual based fear conditioning is more hippocampal dependent, while the cue-based is more amygdala dependent. So although we don't see the same difference in the cue-based, it could be because of different brain regions. And so once again, I have taken the EAE clinical score for each of these mice throughout the whole study. And for these overexpressor mice, there doesn't seem to be a difference between the two groups. But again, this is due to different mechanisms. So this could be further investigated. And also I need to add more mice to these groups to really look at the differences. Okay, so then I also looked at the correlation between the freezing percentage and the score for the at chronic EAE. And once again, I didn't find any correlation with the disease score and the cognitive outcome. So this reinforces the idea that the cognitive outcome and the locomotor outcome are due to two different mechanisms. And so far with my AIM-1, overall, although more animals are needed to confirm these results, this data indicates a trend of a worsening of cognitive deficits in the knockout mice compared to the controls, and an improvement in cognitive function in the overexpressor mice compared to the controls. And so although this data is still preliminary, it supports our hypothesis that Astroglial TNFR2 is protective against cognitive dysfunction in EAE. And so in addition to these behavioral tests, I've also collected tissue from each of these mice in the process of looking at the expression of synaptic proteins and glutamate receptors, as well as analyzing dendritic spine morphology and density, which will give us a better idea of what role Astroglial TNFR2 is playing in synaptic function. And so this brings me to my second aim that I'm going to briefly touch on. This is something I've just started. And so although it's important to look at the role of Astroglial TNFR2, but we really want to understand the mechanisms by which Astroglial TNFR2 is working to make changes in cognitive function and synaptic function. And so that's why for my AIM-2, I want to investigate the epigenetic mechanisms that underlie Astroglial TNFR2 function and synaptic plasticity and cognition physiologically and following EAE. And so epigenetics has been shown to play a role in cognition. So over the past decade, a growing body of evidence has underscored that epigenetic mechanisms can regulate complex human processes such as cognition. And studies in animal models support the view that epigenetic processes play a role in learning and memory consolidation, and they can help transmit acquired memories even across generations. And epigenetic mechanisms have been shown to impact gene transcription, protein synthesis, and synaptic plasticity, which can lead to effects on learning and memory and other cognitive processes. And so for this aim, I hypothesized that Astroglial TNFR2 regulates synaptic function and plasticity, and thus cognition through specific epigenetic mechanisms that fine-tune the expression of genes involved in neuron to glia communication. And so for this aim, I'm using this low input multi-omics approach. So I'm working with the Louis Tuesta lab, who has made this really nicely working cut and tag system where you really only need a low input of cells. And basically our goal is to correlate astrocyte specific changes in gene expression using RNA sequencing with changes that occur in the chromatin architecture using cut and tag. And we're particularly interested in looking at histone post-translational modifications that can occur in response to an upregulation or activation of TNFR2 in astrocytes. So how this is going to work is we're basically going to dissect the hippocampus from naive mice as well as EAE 25 and 40 DPI, and we'll create a single cell suspension. We'll sort out the AXA2 positive astrocytes. AXA2 is a marker for astrocytes. And then these purified astrocytes will be split for RNA extraction or nucleation with light cross-linking. And so then with the help of the Tuesta lab, we'll perform cut and tag as well as RNA sequencing to give us a data set where we expect to uncover astrocyte-specific changes in histone composition that can regulate the transcription of genes that are involved in synaptic transmission upon TNFR2 overexpression or overactivation. So overall, the significance of these studies, we really want to expand the body of knowledge on TNFR2 signaling in the CNS and its role in MS. We want to uncover the molecules and pathways regulated by Astroglial TNFR2 that are responsible for its role in modulating cognitive and synaptic function in EAE and MS. And ultimately, the goal would be to uncover a therapeutic strategy for controlling or counteracting cognitive decline in MS. So with that, I just like to thank my lab, of course, Roberta, my mentor, and all of the past and current members, as well as my committee and collaborating labs, the graduate program, and all the cores that help us out so much. So thank you, and I'll answer any questions.
Do we have any questions?
Oh, hi.
Thank you for your talk. It was really interesting. In relation to why you're going to explore females, is there a specific reason why you're particularly interested in females as opposed to continuing the work at just looking at males?
Yeah, so in the EAE field, females are mostly used, and that's mainly, I think, because, MS manifests more in females than males. It's a 3-to-1 ratio. And I had already been doing work with females, so I wanted to continue that. And then also, I saw in such as the novel object test and the fear conditioning test, there weren't differences in the naive conditions. And so that makes us able to look more at what's happening, what's disease dependent in the EAE conditions when they're the same in the naive conditions. So that's why I wanted to first investigate females, but I'm also in the process of doing males as well. And I'm really interested to see if there is sex differences in EAE and what that could be from.
Thank you.
Mm-hmm.
No, it's the broadcast.
Hi, thank you.
Hi.
Very nice talk.
Thank you.
I'm just curious, why not just use a drug to activate or block the TNF receptor 2?
When we can do this specific upregulation, specifically in astrocytes, we can, if we use a drug like a TNFR2 agonist, it's gonna go to all cell types. So this allows us to look at a cell-specific effect, which is what we're interested in. So yeah, that's the benefit of using those overexpressor mice for the cut and tag and the RNA sequencing, because we can look specifically at what's happening with the astrocytes.
Yeah, because when they use anti-TNF alpha drugs
Mhm.
to treat RA, for example, they can produce MS symptoms. So that's a problem with that particular blocking drug.
Like when it blocks everything. Yeah.
Yes, it blocks everything.
Yeah.
Any other questions? Zoom listeners. Oh, we have Dr. Dietrich here. 1 second.
Nice talk. Thank you very much. Are you going to look at any evidence of demyelination in the hippocampus?
Yes, that's definitely something I want to further look at. So far,
I mean, epigenetic,
yeah.
That's great, but suppose it has nothing to do with demyelination. Suppose it all has to do with inflammatory cascades targeting synaptic function, which we know all about, which we know something about.
Yeah, I definitely want to further look at that. Yeah.
I'm really interested in this because I used to think about demyelination in MS and other situations as robust demyelination, but now we know that there can be very subtle defects in the myelin that have major effects on axonal conduction and all this other things. So anyway, so it's interesting to you, you might look at it. The other thing has to do with I mean, it'd be great to do some really nice high resolution confocal microscopy in the hippocampus or cortex and see exactly if you see any evidence of subtle changes. The other thing is that I've always been intrigued by, people and animals that have spinal cord injury and you have cognitive problems. And we might blame it on, well, that subject had, both head injury or a cervical injury and whatever, but not in the rat models. They're publishing studies now. So I always thought that what's the mechanism for like spinal cord injury having an effect on cognitive function? And again, it could be circuit dependent, but it also could be just inflammatory cascades up and down the neuroaxis. So anyway, I think it's a very interesting area of research.
Yeah, it's definitely interesting. And it's something that's not really looked at. It's definitely a major gap in the field. So, yeah.
I'm going to flip Dalton's question around and ask it in the opposite way. So what do we know about injury to MS patients at the level of the synapse in the spinal cord? So one can say the obvious measure is looking at some type of imaging. You see demyelination lesions, you see probably axonal problems, and you make assumptions that a lot of the functional deficits are just due to that. But is there evidence that there is any direct effect on synaptic function, independent of those things? Because that in itself could be something that these studies may or may not kind of allude to, saying there could be actually multiple targets that MS is affecting that isn't as obvious to, what we've looked at so far. So I just want to put that out there.
Yeah.
Maybe.
Definitely.
Any other questions? Our Zoom listeners, do we have any questions? Unmute yourself and just shoot. We have one here.
Awesome talk, by the way.
Thank you.
Question about your original study where that led to your follow-up studies. You said that they had removed the TNFR2 or TNFR1 and then reintroduced it, right? So the animals that you were comparing it to, that you ended up seeing the increase in or the increase in the declining of the cognitive function, were they just looking at animals that had the TNFR1 removed and not reintroduced, or what was the group that they were comparing it to?
They were comparing it. So this was the way they re-expressed TNFR1 was with tamoxifen. And so they were comparing it to a group that just received oil instead of tamoxifen, as well as a group where TNFR1 was knocked out in all cell types. And so compared to those two groups, the one that had TNFR1 re-express had less freezing behavior. So meaning it had some cognitive deficits. Yeah. So it was compared to two control groups, one that was like a transgenic, but had oil instead of tamoxifen, and one that was where TNFR1 was knocked out in all cell types.
Okay.
Yeah.
Okay. Thank you.